Andre Bachmann, who many of you have known before, comes to us from Hawaii. He is an assistant professor and the chairman of the Department of Pharmaceutical Sciences, did I get that right? And the College of Pharmacy at the University of Hawaii in Hilo. He's going to talk to us today on DFMO and where we are today. I can put your talk in here. Let's see, no, that's not yours. Is yours up there?
It was.
Did they load it? What is it called?
NMTRC Symposium. Right, I want to.
This one?
Yeah.
Okay. Great. Yeah, sure.
All right. Well, thank you very much for this nice introduction. Thank you to Dr. Ashlyn and Dr. Scholler for inviting me again to this conference here, which I truly believe is a unique conference in that it brings together researchers, physicians, and parents and advocates, which is something that one does not find probably in any other setting, and I think that's why it is such a successful consortium. I should say I'm not the only one from Hawaii this year. We have Dr. Randy Wada from the Kapiolani Children's Hospital from Honolulu here too, so he had the same burden as I have to travel all the way from Hawaii to come here, so it's great to have you here as well. So what I will try to do today is to just very briefly give you a background on what the DFMO and ODC and polyamines are all about. I have talked about this before, of course, and some of you have heard about this as recent as last week in Miami at the ASFO, but I hope I will have some new things for you to show as well. So for me, this story started really back in 2001 and more directly in 2002 at the ANR in Paris when I attended the conference for the first time, the ANR conference, and was pretty new to neuroblastoma, and I realized at the time that NMIC amplification, as you all know here, is very important in this disease. And to me, downstream of NMIC was this enzyme called ODC, or ornithine decarboxylase, which is directly activated by CMIC and NMIC as well. And so I was thinking at the time that, as I will explain in a minute, the polyamines are important in cancer. If we could use that drug DFMO, or difluoromethylornithine, also called L-ornithine, in a neuroblastoma setting, would be wonderful because it's already available and has been already used in different settings, as I will tell you in a minute. And that was just a thought, an idea, a hypothesis back then. And as you very briefly will see through the preclinical work, we progressed then to the phase 1 study after I fortunately met Dr. Scholler at the conference, and to now a phase 2 study, which I will also briefly mention at the end. And finally, at the very end of the conference, of the presentation, I would like to show you some of the more recent data that we generated with a new combination with DFMO called AMXT. So I learned two things at that conference. For me, it was the first time I was introduced to parents that had children with neuroblastoma, and as a basic scientist, that was not something that I'm exposed to every day. First, I realized ODC, even though it has been very well known at the time already that polyamines are important in cancer, ODC is a novel therapeutic target for MYC-driven neuroblastoma. And also importantly, I realized that neuroblastoma, again, is not just a cell culture model system, and again, to this audience, that is obviously true, not something new, but for basic PhD scientists, oftentimes they sit in the lab and they just look at cells under the microscope and they forget that this is truly a cell line that comes from a tumor that at some point most likely had killed a child. And so this is a true important model that we work on, and I make that clear to the students every year, and I bring it up every time when I give a talk, that we should never forget about that. So what are polyamines? They are originated from a molecule called ornithine, and this enzyme ODC does convert it to a diamine called putrescine and further convert it to spermidine and spermine. These are collectively called polyamines, and ODC is, as I just mentioned, activated by MYC-N and also c-MYC. And when it is activated, it activates the enzyme, and these polyamines are overproduced in tumor cells, which helps the cells to hyperproliferate. And if we now block this enzyme with this suicide inhibitor, DFMO, we are expecting the polyamines to be reduced in the cell, and as a consequence, the cells go into arrest and the proliferation is stopped. This is oversimplifying the model, but this is in essence what we're trying to achieve. So DFMO is not a new drug. It was, as I said, used already in other clinical settings, and it was, in fact, developed back in 1978 by Merrill Dow. And there's a long story behind DFMO, which I will not talk about today, but the bottom line is it was very successful in treating African sleeping sickness and was used, or is still continued to be used by the WHO, now in combination with nifurtimox, which is in complete coincidence with the work that Dr. Scholler does with nifurtimox, but it is used as a NECT therapy, they call it, in combination with DFMO. And it is available in IV formulation as well as oral formulation, as it is being used for neuroblastoma patients. And as we probably all know by now, DFMO has very low side effects, even at quite high doses, which is very unique. And currently, now it is being used as an investigational drug in the neuroblastoma patients. This is just showing quickly how the African sleeping sickness works. It's a trypanosome, a parasite that is transmitted by the tsetse fly. And this little bug here uses ODC as well. And because of the DFMO that kills ODC in the trypanosome, the disease is stopped in progression afterwards. So this drug had been used at the time for many years before we started to look at it for the use in neuroblastoma, and these are some from my very first data that I had produced in the lab back in 2002, just showing that different cell lines were effective in treating responding to treatment with DFMO in a dose-dependent manner. And I'm not going to discuss any preclinical data today, but I just wanted to show some pictures. This is two neuroblastoma cell lines, control cells, untreated, and here treated with DFMO. And if you add back into the cell culture plates this polyamine spermidine, you can see that the cells recover again, clearly showing that the depletion of spermidine and putrescine by DFMO was the reason why these cells stopped growing. As we're adding it back in, they start to grow again, and similar to the controls. So many years of research then, we investigated in detail what exactly is happening to the cells on a molecular level, and I'm not going to show you all these data, but I'm just summarizing it here with two papers that we published. We basically showed that P27 was a key protein upregulated in response to DFMO, which through RB then activated the G1 cell cycle arrest in those cells, and we further studied P27 and also AKT in that cell setting a little bit more, and so basically laid the foundation for what is going on in neuroblastoma in response to the treatment with DFMO. And then a very fortunate thing happened to me at a conference in 2008 in San Diego at the AACR. I was actually introduced by a parent talking to me at the poster session, introduced to Dr. Scholler, and he brought us together. And at the conference, Dr. Scholler was at that conference too. And this is where the story for the clinical side started. And Dr. Scholler was very interested in DFMO, and so we started immediately to collaborate thereafter. And Dr. Scholler's lab then performed some in vivo studies with xenografts, here shown in control, and then treated with DFMO and etoposide. And we had tested all kinds of different combinations, but at that time we felt that DFMO and etoposide was the best combination also to further pursue for a clinical trial. And fortunately, there were two other groups that independently had interest in DFMO and looked at a transgenic mouse model at that time, the TH-MYC mouse model. Both John Cleveland at Scripps, Florida at the time at St. Jude's, and also Michael Hogarty from CHOP showed in two independent studies that DFMO was quite effective in those neuroblastoma mice. So that was great independent support towards our phase 1 trial that Dr. Scholler, obviously, as you all know, conducted over the last several years and through the Neuroblastoma and Medulloblastoma Translational Research Consortium. And so the phase 1 study was a safety study for the refractory and relapsed neuroblastoma with DFMO alone or in combination with etoposide. And the trial opened in February of 2010, less than 2 years after we had met. And the primary objective was to determine the safety and the tolerability and maximum tolerated dose of the oral DFMO as a single agent, as well as in combination with etoposide in pediatric and young adult patients with refractory or recurrent neuroblastoma. And the secondary objective was to look at the activity of DFMO as a single agent, as well as in combination with the etoposide, and look for progression-free survival, as well as overall response rate. We also then planned on looking at various pharmacokinetic parameters and do some biological studies, like assessing the polyamine levels in cells as well as in urine samples, as you will see in a minute, in pre and post-treatment situation. So, and I should have changed this here. Actually, Dr. Gerner, who's here in the audience, has done most of that work. So how did the clinical trial take place? This was an initial evaluation phase, and then the first cycle was DFMO by itself, twice daily for 21 days, and there were 4 different levels starting at 500 milligram per square meter. There were 3 patients enrolled at that level, and after the first 3 patients were evaluated, then the study moved to the level 2 to a higher dose range. Etoposide stayed at the same concentration and was added into the treatment plan after cycle 1 for cycle 2 through cycle 5, again every 21 days. The etoposide was only given for 14 days, though, at 50 milligram per square meter. And so that was the general setting. And so just in April, this poster was presented at the AACR in Washington, D.C., and the title was a phase 1 trial of DFMO as a single agent in combination with etoposide. Since the primary objective of this study was to look at the safety, I'm showing you here one figure coming from the poster directly, showing all the different adverse events that have been observed. There were mostly grade 2 side effects, as some anemia, and I'm not going to go through the entire list here, but there was 1 grade 3 and grade 4 level anemia. And so the side effects were relatively mild, as was expected from all the experience that one had from previous trials and from the situation with African sleeping sickness treatments yes. So the response data are shown here, and the progression-free survival. So there were 15 out of 17 evaluable subjects had stable disease through at least 2 cycles of DFMO. 1 subject had a complete response to their bone marrow disease, and 2 of 17 subjects were stable for over 1 year, and 2 were actually stable for over 3 years. And 1 of the 17 subjects are currently on the study, or continue to be on the study at this point. Progression-free survival, the mean PFS to date is 187.4 days versus 42 days for historical controls. And those with stable disease had a mean PFS of 221 days versus 119 days for those who had progressive disease with the initial 2 cycles. So that's in summary what the study found, and I'm sure Dr. Scholler is able and willing to discuss this in more detail, what exactly the patient responses were. This is showing a PET image here, the PET improvement of one of the patients here after, or this before DFMO treatment, and this is after DFMO treatment at level 3, which would be 1,000 milligram per square meter twice daily. So quite a nice improvement, as you can see, compared to the situation before treatment. Now, these are all data that were generated by Dr. Gerner at the University of Arizona. These are polyamines that were measured in spot samples of the urine from patients. And this is a pretty busy slide and a lot of numbers, but these are some of the polyamines that were measured in the urine samples. Diacetylspermine, histamine, monoacetylspermidine, puracine, and spermidine. These are the 3 traditional polyamines. And basically, key was here that puracine in the responder group significantly was reduced over these 1, 8, and 15 days, versus the non-responder group did not show these kind of effects. And some other things were found now that I have to say there needs to be more done, of course, to really solidify these data, but this is a nice first stab at what could be potentially a marker for patients later on. Monoacetyl spermidine decreased also in the non-responders and also in the responders, and histamine increased in, it decreased in this situation as well, yes. So, and spermidine, there were not any significant changes. But the bottom line is, I think, from this data that certain polyamines may serve as a predictive marker in the urine of neuroblastoma patients, and we will continue to look at polyamines in the urine samples just to further study and see what exactly is going on and if they can be used as markers. So the biological active dose that then was determined was 1,500 milligram per square meter POBID, and this is the range that we had tested in this phase 1 trial. Prolonged stabilization of these was seen with clinical benefit in greater than 50% of the patients, and again, urine polyamines appear to be a marker for response to DFMO therapy. At least some polyamines seem to respond, and the biological correlates will be further explored in a phase 2 study, which is currently underway, and about which I will just now for a few minutes talk to you about. Now, the idea of using DFMO in a chemopreventive setting came really from this very interesting study that was performed by Dr. Maskens and Dr. Gerner, who is here with us in the audience, in a different cancer model, in the colorectal adenomas. And this is a phase 3 study that was conducted with DFMO in combination with an NSAID called Sulindac. And they had remarkable findings in this study showing that all adenomas reduced by 70%, and advanced adenomas were reduced by 92%, and the recurrence of more than one adenoma was reduced by 95%. So this was amazing. Now, the setup of this trial was that patients received DFMO and Sulindac daily for over 3 years, and they had all polyps removed before treatment with DFMO and Sulindac, and then after 3 years, they basically looked whether or not these patients had or did not have any occurrence in colorectal adenomas. And it was, the placebo effect was obviously at the control for this study. And so this was in the chemopreventive world a milestone achievement, and so we thought that for neuroblastoma it would be nice if one could now look at patients that are in remission and give them DFMO on a daily basis to see whether we can keep those that are at high risk for relapse, keep them in remission for a longer time, or hopefully for forever. So this trial then again, Dr. Scholler opened the trial just this or last year, it's already 2013, in June 2012, and it is offered at 8 NMTRC hospitals currently. So the primary objective of this trial is to evaluate the preventative activity of DFMO as a single agent in patients with neuroblastoma who are in remission based on event-free survival or overall survival. And the secondary objective will be to continue to determine the safety and tolerability of DFMO as a single agent in that patient group. And we will also continue to look at the biological correlates and study polyamine levels and ODC activities and so on in those patients. The DFMO is an open-label, multi-center study for patients with neuroblastoma in remission, as I said. It will be done over 24 cycles. And in this study, the dose range is from 500 to 1,000 milligram per square meter for 28 days, and there will be 2 strata. Stratum 1 will be subjects that are in remission at the end of upfront therapy, and stratum 2 are the subjects that are in remission after any previous relapse. And I think the numbers are here for stratum 1, about 40 subjects, and stratum 2, about 32 subjects will be enrolled. And I think currently the status is that already 30 patients, or maybe more by now, have already signed up for this study. So, in summary, I basically tried to summarize about 10 years of work, but the preclinical preclinical work done in my lab was telling us that DFMO seems to induce P27RB pathway on the molecular level, and by doing so arrest cells at the G1 phase. And we then found in mice in vivo, and also others showed concomitantly that DFMO is effective in xenografted as well as transgenic mice models. And that supported then our attempt to then perform this phase 1 trial, which now just ended in I think, December 2012, to do a phase 1 study with DFMO with relapsed and refractory neuroblastoma patients, and finally led then to the phase 2 trial that I just presented to you using DFMO in a chemopreventive setting for patients that have high risk for relapse. So what's up in the future? We're continuing to work towards finding new combinations that are effective for further depleting polyamines. We have focused on one inhibitor more recently called AMXT. It's a polyamine uptake inhibitor. Sometimes cells, when they feel that the polyamines are depleted inside the cell, they start to pump in from outside polyamines. And so what one can try to do is block those pumps and thereby further depleting the polyamine uptake. And AMXT is such an inhibitor. And combining it with DFMO, we are hoping to basically deplete now the polyamines intracellularly with DFMO and the uptake extracellularly with AMXT. And just one figure from a recent paper from Dr. Scholl's lab showing here continuous measurement of cell growth. These are cell lines. These are not mice studies, but nevertheless showing that the vehicle here by itself, the cells grow. DFMO by itself does inhibit. AMXT by itself does inhibit. But look, the combination together clearly showing a synergistic effect in the cell culture. And this, again, continuously measured cell growth over time. In vivo, there was also a big change as shown in these PET-CT images. Here, the control showing the tumor mass. Here with AMXT, we find small decrease. The DFMO definitely also decreases the mass, but again, combined, the two together had the strongest effects and probably synergistic effects. And so we're hopeful that this combination will be further studied and maybe will lead to a clinical study as well. Some of the data that I just showed were recently published. Actually, it's in press. I don't think it's actually out yet, the paper, but this is coming to you shortly. So other projects that are ongoing in my lab are identifying some improved delivery systems for DFMO and other drugs to the neuroblastoma tumors, and one of the assistant professors in the department that I'm in works with nanocarriers, and so we're working together on producing some new type of nanocarriers that engulf DFMO and etoposide or Velcade, anything you want to put in, basically, coupled with antibodies against GD2, so that we have basically a tool to bring the drugs closer to or faster to the tumor, and also hopefully in lower doses. But this is in the development stages, so we are just trying to basically figure out if there are such ways of increasing the efficiency by which the drugs reach the tumor site. Then some genetic epidemiologic studies are planned, for example, looking at SNPs or single nuclear polymorphisms in ODC genes. There has been, again, in other types of cancers like adenomas, shown that some genetic mutations in ODC genes correlate with prognosis or outcome and risk of recurrence also, as well as the efficacy of DFMO. So I would like to look at patients on the genomics level and look at whether ODC has any mutations and if there's any SNPs present in this particular gene, and also some other polyamine-related genes like anti-Zyme and so on. And finally, this is, I would strongly underline this, a hypothesis, and as it is true for other types of cancer viruses, and even like in the case of H. pylori, bacteria have been shown to have an effect in cancer, and as far as I know, in neuroblastoma, nobody has ever reported anything like that, but I strongly believe that, at least in some cases of neuroblastoma, there might be some situation where viruses, maybe during pregnancy or even later on, that they may play a role in developing neuroblastoma. And there's some really brand new technologies like mass specs, abbreviated as LASI mass specs, that would allow us to search in those tumor samples for potential presence of viruses that could have at some point induced cancer. I just give you one example. Cervical cancer and human papillomavirus led to a Nobel Prize in 2008, and that was a long discovery in the making, but Dr. Zurhausen showed quite nicely that viruses do cause cancer, and in this particular case, cervical cancer. But this is a true hypothesis. I have absolutely no data or anything to say, but I just think that it's worth looking for it, and this is something that I'm focusing on also in my lab at this point. So finally, I think I used up all my time. I would like to thank my lab. This is now at the College of Pharmacy in Hilo. And of course, over the years, many people have worked on this topic that are no longer in my lab. Dr. Como actually is an assistant professor herself now in our department, and many students that came and left thereafter worked on this project. I was very fortunate to work with many collaborators, first and foremost Dr. Scholler, Dr. Meskens, and Dr. Gerner have been very helpful during the years and in helping us with our studies, and Dr. Feith and Dr. Peck from Penn State University, and a whole range of other investigators, mostly PhD scientists that have contributed to this work. I was fortunate to get funding from the NIH, and I hope I will continue to get funding from the NIH. As we are in the renewal phase right now, and we'll only find out soon. But the Hawaii Community Foundation is also a great place that has supported with several grants this work and some other work that I do on proteasome inhibitors. And I've been very fortunate to have support from them. Magic Water Foundation has also supported our work, so we're very grateful to that. Cancer Prevention Pharmaceuticals provided DFMO in the phase 1 study. And as Giselle said at the very beginning, all these parent organizations are extremely important in this line of work. And on behalf of Dr. Scholler, I also would like to thank all these organizations for contributing to the NMTRC. With that, I'm at the end of my presentation, and I take any questions that you may have. Thank you.